Hello, everyone, and welcome to our series of webinars. Today, I would be talking to you about some updates on the vegetable systems trial, the soil health and vegetable nutrient quality. I'm Gladis Zinati, the director of the vegetable systems trial at Rodale Institute, and I am very happy to give you updates on in the past few years, what we have in the vegetable systems trial, and some information how we can use the results that we gained throughout the years. To start with, I would like to do some housekeeping points. You can hear me. If I am not audible to you, make sure you contact Justin Barkley, who is at 610-683-1442. And also you can use the audio settings on your screen. Please, for your questions, I would appreciate that you put it in the QA button and list your questions there, so we don't get disruption if you put those questions in the chat. At the end of my presentation, I would be open the floor for questions and answers. And recording is in process right now, and you will receive a link, or you can access the website in a week or so for this webinar. I want also, before we delve into this webinar information, I like to thank our funders, mainly the Grantham Foundation, the Department of Agriculture, Pennsylvania Department of Agriculture, the Orange County Community Foundation, Fertrell, also the research that is funded by the Pennsylvania Growers, the Marketing and Research Program, and mainly Rodale for hosting this work and working on a bigger picture of solutions for the world. Just to give you an outline so we can know what we will be covering today. It is a quick background of the VST, or the Vegetable Systems Trial, especially because we have new attendees, and I usually provide a few slides at the beginning so everybody would be on the same platform. Also, I would be covering some highlights of the results, mainly would be about the microbial community functionality, mycorrhizal fungi root colonization, and nutrient quality index. I selected certain crops to give you examples. Also, what we have current projects at the moment, and certain outreach educational programs that I have been involved and will be involved in 2023 in the first few months of the quarter. Then we open the session for Q&A. And for me to be better focusing on this presentation, I would turn off my camera at the moment, and then I will open it at the end when we have the question and answer session. A little background here, mainly in the past 4 or 5 decades, we have seen concerns arise, and more we see the American diet has become energy rich and nutrient poor, filled with toxic chemicals and synthetic materials, that it is becoming an issue. And we have seen, as in published papers or in real life, increased rate of chronic diseases and reduction in the immune system in adults and children. Those could be attributed to many factors, mainly in the past 5 decades or 7 decades. It is due to the intensive work that's been done in the agriculture by degrading the soil due to intensive tillage, depleting the soil from its nutrients, reusing that soil with depleted nutrients and adding more of the synthetic fertilizers to make up for the yield, and toxic chemicals that became built up into the soil, make it unhealthy. And To add to this problem, the breeding programs that have been during this industrial era for fruits and vegetables, they increased the yield and size of the fruits on the problem that really affected nutrient quality and density. And in that case became a problem where we found in published literature showing that about 38% reduction between 1950 and 2000. What it means to us, meaning there was a dilution effect here. And so in the past, before 1950, if you eat one broccoli, that will satiate you for the 1200 milligram per 100 grams of calcium. And calcium is a very important mineral for our growth and development and sustainability. Now, if we go and assess broccoli, it has 1/3 of that amount that we require for the daily value, meaning that we really need to bulk more, to eat more, to reach what we require for that amount of reference daily value. So this brought us to think properly and think how we can solve such issues. And as you know, Rodale is the pioneer for looking into innovations, ideas, and how we can look for solutions. And so in 2013, when I was working and looking into solutions for vegetables after coming back from many meetings I had with the growers, especially the vegetables growers, I realized that we really need to help the vegetable growers because there is plethora of research is already done on grains, but not on vegetables. And so in 2016, we hit the ground running and we established on 2.6 acres a long-term trial where we are comparing side by side organic to conventional. And in these systems, we are looking into how the cropping system and management practices really impact the soil health by using these practices, and how those will affect the environment and plant nutrient density, because we know at the end, we are all consumers and it will be affecting the human health. The vegetables that we selected included beans, lettuce, sweet corn, winter squash, and potato, because these represent the tubers, the fruits, and the leaves. So coming back to highlights for the results, we could say that during the 2019, and if you go back to previous webinars I have given on VST, we collected soil samples in cores, and then we sectioned them to different sections. And in previous webinar in 2020, I talked about some of those nutrients and results that we had seen between those management practices and cropping systems. And today, I'm going to give you an update mainly on the microbial community diversity and functionality. This work is a very big and deep because it really requires a lot of work. And during the COVID time between 2020 and 21, really hindered us to provide these results very quickly. But today, you will see a very dense information that I will be sharing with you. And it is really interesting to see these results that we gain information from. And so here, in collaboration with Dr. Janjun Khan, who is a soil microbiologist, and he is from Strauss Water Research Center, we looked into the microbial composition, diversity, and functional genes, and we are going to see if those will translate to the vegetables such as winter squash for yields, nutrient quality, and soil health parameters. And today I will be talking about the first item, the composition, diversity, and functional genes. In brief, after taking the soil samples, we cut them and they do the genomic DNA extraction at Dr. Khan's lab. And then we assess using PCR and qPCR the functional genes and we quantify them. And then we look into sequencing and then applying bioinformatic analysis and statistical analysis for composition and diversity. So let's look at the first result. And here I will be going pretty slow at the beginning, so we all can be on the same page. So what NMDS plots of bacteria archaea community structure for squash across the 3 depths, 0 to 10, 10 to 20, 20 to 30 centimeters. So let's explain what is NMDS. NMDS is non-metric multi-dimensional scale. It is used to evaluate the diversity of these community structures. So on this, on the right, you will see the treatment and depth. And so if you see the orange, it would be the conventional with black plastic. If you see the red, it would be the reduced tillage for squash in conventional. And if you see the green dots, these are the organic black plastic. And in comparison, we have organic reduced till. It is in the dark blue dots. So what does this graph tell us? It tells us whenever we are looking at these points, these are multiple hundreds of points together, but these are telling us about the communities, especially the bacterial archaeal communities, that how far they are from each other and whether they are different from each other. And you could see here that you can separate with a line these communities that are in the conventional from those in the organic. And mainly you can also see that those in the green and the blue, they are statistically different, and they are different from all the other ones when you compare these blue to the green. And also, as we go with depth from 0 to 10 all the way to 20 to 30, you could see the distance is getting further and further. And so meaning it is more, these structures are different from each other. So this is for the bacteria archaeal in squash at different levels. So let's look now at the fungi. For the fungi, for the same scenario for squash, you could see that also that there is a difference between organic and the conventional, pretty good. The statistical is really good in that case, and the communities are far from each other, meaning they are different. And it is more, you could see those when we go into 20 to 30 in the fungi or fungal communities. Let's look at potato now. For potato, same scenario, we are using the same colors. And for the bacteria archaeal, you could see that the, should be here, potato, sorry for that. It is significant only between organic and conventional, and at 20 to 30 versus the other ones. Again, sorry for that. It should be here to be potato. And so you could see here that they are different in the organic versus conventional, and with depth as well. So this graph is talking about the ammonia oxidizing archaea. Archaea is one of those microorganisms. Long time was considered as a similar to bacteria, but that term became more specific now, and archaea, they are the prokaryotic. And that means they don't have a nucleus as the bacteria. And these are favorable in conditions when soil is into acidic near to neutral, when the soil is low in ammonia concentration, and when there is presence of oxygen and cycling of nitrogen. So meaning that wherever we see more of these copies of the genes that it is highlighted here. So what you see in blue here, it is organic reduced still. What you see in the organic black plastic in green, and the conventional in the pink and the orange. So what it says here that more of those copies of genes of the ammonia oxidizing archaea were found in the organic reduced still, meaning they are more abundant there to cycle the nitrogen, and they play a key role in the nitrification, meaning oxidizing of the nitrogen to nitrite and to nitrate. The shadows of those colors, meaning that the darker the shadow in each of those, meaning that amount or abundance is lower. So mainly you will see them into the 0 to 10 or 10 to 20, depends on the practice. And so these are important, and you can see the number of abundance here is pretty high. And let's compare it to the bacterial one. So on this one, you will have a bacterial, and you could see that the abundance is much lower than the archaeal, which was on this side. And there was no significant difference here. So they are all the same at all levels, but they decrease with depth as well. When we look at the ammonia oxidizing archaea in the potato, we see that the same thing, the organic system, irrespective of it is black plastic or reduced still, the abundance here is more than in the conventional, which is really good to see that. And then it is showing that they change with depth, and you could see as you go in depth and darker colors, they are less and less. If we look here at the bacteria, the AOB, the archaea oxidizing bacteria, you could see that only the organic reduced still in potato showed more activity, but nothing different across the other treatments. And again, as we go with depth, that number of genes starts to be lower and lower. So a summary for this part of these results, it shows that microbial community structure differ with cropping systems and tillage in squash, whereas only differs with system in the potato. Also, the microbial community structure differ mostly at deeper depths, meaning as we go deeper and deeper, they will be different from each other. As for squash, the ammonia oxidizing archaea were abundant in the organic system, especially in the reduced still, indicating more nutrient cycling and availability to plants when we are practicing organic system with regenerative tillage. Same as the depth goes deeper, these AOAs will decrease, and they were proportionally greater than the ammonia oxidizing bacteria. There were no difference across the bacterial systems for squash. For potato, the difference we saw, it was only when we looked at organic versus conventional. It was more availability of those genes of bacteria and, archaea, and that really helped to improve cycling of the nutrients to the plants. The bacterial, so the ammonia oxidizing bacteria were greater in the reduced tillage organically in the potato. The other project that I will give you updates on, it is about a project we started in 2021. And that's in collaboration with Dr. Wade's Heller lab, Dr. Wade Heller and Dr. Joe Carrera, his postdoc. We are working on a 2-year project about crop root colonization and mainly and speciation to look into how that impact the nutrients and the colonization when using arbuscular mycorrhizae from outside, meaning not naturally done, but it is by introducing arbuscular mycorrhizae fungi. Just for those who do not know what's arbuscular mycorrhizae fungi, which is termed AMF, mycorrhizae or mycorrhiza means the fungus root, and arbuscule means a small tree. And so you could see here, these are the spores that they carry to make more of the stretches, or mycelia we call them, and the arbuscules would be inside. And so there are small trees inside the branches that these spores make to connect with the plant root. If under the microscope, we see the roots like that without all these arbuscules and hyphae, we know that there were low root colonization. So let's hop into the results and see what we have found so far. So for sweet corn in 2021, we start the seedlings in the greenhouse, and this data showed that between organic and conventional, they may have varied across the treatments here, but they were between 2% to 12% maximum root colonization. That because the seedling is about 7 days old only. Then we transplant them into the systems in the vegetable system trial, and we planned them into a satellite experiment where we looked into a reduced tillage versus till. And so here at harvest, so if we look at the green bars over here versus the red bars, the red are the conventional and the green are the organic. And if we look across all the greens only, we see no differences. And if we look across most of those red ones, there is no difference across the treatments. However, you could see that at harvest, definitely we saw more of root colonization and increased from about 10% to about 45% in all of them on average. And so you could see more of the root colonization enhanced in the organic over the conventional. So let's look at now the tillage effect. And in this case, the green bar here is the reduced tillage. And here the harvest at harvest, which is the till in the red. Again, we could see that at the green ones, the green bars are greater in some cases, such as in these two treatments where we use the inoculum called PA127A and ON2205B. They showed both of those are greater and enhanced root colonization in the reduced till, where we rolled crimped the soil, the cover crop, and planted into it, than tilling and using multiple tillage cultivations during the season. It seems those two inoculum are more sensitive to tillage compared to the VSD-ORG. VSD-ORG is an inoculum that we started from the vegetable systems trial, and we wanted to see in comparison to those that we introduce if the tillage will be effective or not. Let's move to squash in this case, and here we have the same scenario. We start the squash in the trays in the greenhouse, and you could see that they are also different. The ones that were inoculated with VSD-ORG had more than the others at the beginning, and less than and range between 2 and 10% root colonization. But when we go to mid-season, and here you could see that the first two bars of each, the dark and the white bars, they are representing the organic, and the ones that dotted versus the gray, these are representing the conventional. And you could see in all of them that mostly that the organic is more giving us at mid-season more root colonization. But let's look at it when we harvest. And so at harvest, again, these you could see that it is mostly now coming to about 45%. And so when we looked into those by species or by treatments, we could say because these treatments didn't vary among the different systems or the different practices, we could combine these two, which is the organic black plastic and the organic RT. These are similar, meaning the variances that we see, it is only because of a cropping system and not because of a tillage or interaction. And so at harvest, you could see that the organic did much better in root colonization than the conventional one. And there was no effect on the tillage. So in summary for this project, most of the root colonization was 45% at harvest, which is really good in both sweet corn and squash. And we saw that the sweet corn is impacted by system and by tillage, meaning that it is better if we can reduce the tillage and improve it by integrating organic practices. In the squash, definitely they did better when it is organic, and the tillage at this point didn't show any effect. However, we are still working on the data of the 2022, and this is very important because we need to see if those change with time. The other project that I wanted to update you upon is about nutrient-dense crops, and let's take a moment here to describe few information. What does it mean? So when we say a crop is a nutrient-dense, meaning the nutrient content in it is greater than the sugar content, meaning that the more we have a greater nutrient content in the crop, meaning it is rich in antioxidants, vitamins, proteins, and minerals. This is very important because as consumers, when you consume nutrient-dense food or diets, we'll have, or we include them in our diets, we will eventually feed our human gut microbiome. The human gut microbiome is the center of all the things that for the enzymes, for the chemical production that we have in our bodies that really affects the functions of all organs. And that's very important. What you eat is what helps you to react to, and that's very important in avoiding any diseases in the future. So nutrient dense, meaning healthy people, because if we include a nutrient dense food in our diets, we can improve our health, especially when it is free from toxic chemicals. Let's do some definitions here. So as we said, it is the nutrient density is the amount of nutrients proportion to the energy or the calories. It helps us to know what are the key nutrients that are limited or deficient in the diet, but it will help us better if we can do that relative to reference daily intake value. So in other words, so if I would like to look into what I get in the sample from the lab and how much of that it would be required per day, it will really let me know if I am getting a better nutritional quality index, especially when I use the information of the calorie that is produced in that crop or that analysis that I'm using. And we know that every day we would require about 2,000 as adults, 2,000 calories. If that calculation was equal or more than 1, this means that crop or that nutrient quality index is really good. If it was less than 1, so we are missing the amounts that we really require for the day. But Let's look at it in a different way now and calculate those based on what we found in potato and in squash. Before we move to the example and the results, I just want to bring to your attention here that the recommended daily intake value for adults vary among the nutrients or the minerals, and you could see we need a lot of it, of the potassium compared to phosphorus or calcium or magnesium. And those in milligram per 100 grams. So every 100 grams of a fruit or vegetable, we will look what we require for that mineral. If we look at the iron from these micronutrients, we see that it is really needed, the iron and the zinc, in bigger amounts than manganese and copper. And those are very important for the health of the brain, the development, your blood system, and these are very important, especially for development of a child or development of an embryo. When a woman is pregnant, it is very important. That's why they usually recommend to have iron and zinc in their supplements. And so these are very important for the health of the brain. And so other vitamins that are important, vitamin C required more than the B6, but in all they are required, very important to be part of the analysis, as well as proteins. So let's take an example here from 2020, and I'm talking about Lehigh potato, which is the variety that we usually use in vegetable systems trial. So for every 100 grams, this potato will give me 367 calories. So we know that usually we need about 2,000 per day, but let's see if I eat 100 grams, what that will help me in looking into minerals and vitamins and proteins. So this table is showing me the system that the potato has been grown in, conventional or organic. And here we could see the minerals, the vitamins, and the proteins we analyzed for. Just to keep in mind, there are many other nutrients can be added, but for every sample we look at, it is pretty expensive. And so we select the most important components that across all the crops that we grow in VST to assess those nutrients. So what we could see here, that all the numbers, the NQI for each of those are beyond 1, meaning they are really good. If we look at all of them, they are beyond 1 or greater than 1, except calcium. And so what it means that potato, the Lehigh potato that we grow in the conventional or organic, is really good in all of them except calcium. So keep that point in your memory so when we go to squash. So potato is really good in all the nutrients to include. If we include 100 grams, it will provide us with everything except the necessity of calcium. It provides a little bit, but not as much. And also to look here that the potassium, which is really needed in big amounts, and magnesium, you could see that it is greater when the potato is, and significantly statistically, when it is grown in the organic versus the conventional, which is really good news. Let's look if the practice of the management reduced tillage versus black plastic is giving us any effect. And you could see that they are still greater in all those except the calcium, but there was no effect showing for the data we got in 2020 for potato, there is any effect of tillage. Let's compare this information of potato to butternut squash. And again, the calories that we gain from 100 grams analysis, it is 360, which is not so much different in energy from potato, which is good. So we can see the advantage of comparing those two. And then when we look here again, in the organic versus conventional, we looked at all these elements again, and we could see that it is really lower in iron, zinc, and proteins. but it is great in every other element that we assessed there. In comparison to potato, if you remember a few seconds ago, I mentioned that the potato was really good in everything except calcium. So here, the calcium in the winter squash is really good because the NQI is, the index is greater than 1. So, and the potato was really good in everything, including zinc and iron and proteins. And that means that if you combine your diet that has potato, such as Lehigh and butternut squash, you are really providing yourself with a complete necessity of these elements, nutrients, because then you are gaining advantages from both the winter squash and the potato. So here you could see also that it is greater in copper when we assess organic versus conventional. And also, despite the NQI is lower in than 1, but you could still see that the winter squash is really good when it is grown in the organic system, and statistically more than when it is grown in the conventional. When we look at the management practice, the black plastic, tilled with plastic, and then reduced till, where we roll crimp and add and plant the seedlings in it, we could see here that the squash is affected by the management practice, by the tillage. It is much greater, and it is really showing we can gain more phosphorus, potassium, and calcium when we implement reduced tillage, which is very important information to know, that how, when we are using reduced tillage, regenerative organic agriculture is gonna help us to gain more of those nutrients, and also for winter squash as well as other crops, or potato or beans or others, or sweet corn. So in summary here, the nutrient quality index for each nutrient may vary with the vegetable, maize system, and the management. And we have seen that potato varied with the cropping system only, but the squash had the impact of cropping system and tillage. Such information is 1 year I'm presenting here. It is important that to look at multiple years for those crops in these systems and practices, because it really helps us to understand how stable, and that information, it's not only 1 year or 2 years, because we know that in crop rotations and with climate, these numbers may vary, but they could make more of the results rigorous when we do that over multiple years. But it is a great beginning here, a great way to have a reference line and look for the coming years for all these NQIs. For the current project that we are looking in VST, it started in 2020 and 2021, and now continuing in the coming couple of years. We are looking about SNAP beans, the green beans versus the black beans in the vegetable systems trial. And so we are looking at different things, mainly soil microbial community, soil extracellular enzyme activity. And those we are monitoring them at initial, meaning at the beginning of the season, mid-season, and at harvest. We are looking about those changes when we have the different cropping systems. We are also working on the fungi colonization and speciation, if they are different in these beans. And also we are looking about the mineral contents in the beans, proteins, amino acids, and ergothioneine. And maybe not many of you know what's ergothioneine. It has gained momentum, this amino acid. It is a rare amino acid that the faculty at Hershey Medical Center in Penn State. They have been working on ergothioneine, on this rare amino acid, and it is not available in all kinds of plants, but they found it available in oats, and they know, and we all know now, that it is not made up by the plant, it is not generated in the plant, but it is starting the generation of this amino acid in the soil by certain fungi. And they also found it in mushrooms. And so soils that have more of the fungi could lead us to understand more of what would be the ergothioneine content in those plants or others. So in the literature, we know there is ergothioneine in the beans, but there were no description of what are the impact of the cropping systems and in comparison to other crop. So we are collaborating with many collaborators to understand those information, ranging from Strauss Research Center with Dr. Janjun Khan and Dr. Diana Vargas, from USDA with Dr. Wade Heller and Dr. Joe Carrera. And from Penn State, we are working with John Ritchie. He is the pioneer about ergothioneine, with other collaborators from Purdue as well, where recently we got a research proposal accepted, and we will start looking into anthocyanins and other phytonutrients with a collaborator from there, Dr. Radivari. So I am so glad that we have been working on this project together as a big team. In the coming years, we will be teasing out slowly some of those information and present them to you and give you an update. Just to bring to your attention here, Dr. Vargas at the Stroud Water Research Center, she is looking at the extracellular enzyme activity, because it is very important that we look at different stages of the growth, at the beginning, mid-season, at the end, how these enzyme activities are changing in these soils that has the different aspects of cropping systems, organic or conventional. And we are looking at 4 kinds of enzymes, because we want to know how those impact the amounts of nitrogen, amounts of proteins, amino acids, and all related to ergothioneine. So usually we take the soil sample, she will put it in a slurry with a buffer, and then she will look into, after centrifugation, using a equipment here to measure the fluorescence against the standards, and using the plate reader, and then we calculate the activity. And that information, after we get 2 years of data, which would be next year, we are planning to have a team webinar next year to give you more information about this project. As far as outreach, I want to update you on the outreach. We had a great field day this year, and I say great because we have been missing to be in touch with people during COVID, and we had many people became very interested in the vegetable systems trial. And so we had about 389 people visiting us on that day, on July 20th or 22 of this year. Also, we had our first regenerative healthcare conference at Rodale Institute, where we invited medical staff, doctors, students, nurses, educators, where we talked about the importance, how we can heal and provide healthcare by using our regenerative systems by looking into the soil, and how we can look into medications by using plant-based, clean, non-toxic fruits and vegetables. And so many of the doctors mentioned that they rarely take any courses about soil or how the plants are grown or anything. But we are starting a nucleus here at Rodale, where we started our first conference, and this is gonna be every year. And so this year was very limited because we needed to start small, and then I'm sure next year you will see more of availability to sign in. Another recent educational publication we had this year that was published on November 17 in collaboration with Dr. Radivari and Janjun Khan. We published a web article about the phytonutrients in butternut squash as they change with storage, management practice, and cropping system. In the vegetable systems trial, if you go to our website and you click on this link, you can find it. Also, it is available in a PDF form. And I would like you also to join me in future conferences like you joined me today. And those would be at the Mid-Atlantic Vegetable Convention. Those who are available in the region can come to that convention. And it would be on February 1st in Hershey, Pennsylvania. And also at PASA conference, where it would be in Lancaster this year on February 9th. I would be talking about more results, and we'll have a workshop at PASA conference, Dr. Janjun and myself. We will be talking about our research in details, about a workshop for 90 minutes. So I want to leave you with few information here that our research here at Rodale, especially this information about soil health and nutrient density in vegetables, had gained momentum, attracted the attention of many foundations and organizations. And our research is the catalyst for change. And I would like to give you and provide you with some take-home messages. If you leave anything today, I want you to learn and practice this information. If you are implementing regenerative cropping systems and tillage practices, you will definitely enrich soil health and crop nutrient density. Excuse me, I need to drink some water. If we look into implementing those practices, we make our soil healthy, because soil is the core for healing our planet. So please, every day, put this message in front of you and think about it and practice it. Take care of the soil, because then we will have a healthy air to breathe, water to drink, especially because our bodies, it's about 78% water. We will produce better food and eat healthier, denser food, and we will have healthy beings, irrespective if it is a human or animal or any microbes. This is very important to take home message that really we need all of us work together. In the past 2 days, I have been attending a conference ran by the Vice President Al Gore. And it was extremely inspiring for me to listen to all the speakers and the information, the conference that was held virtually. And it is about climate underground. And it makes more sense that our research now that we are doing at Rodale, it is like inside, it is just deeping into that information we all looking for to improve our planet. And with that, I would like to thank you all, because all of us working together, whether we are educators, medical doctors, scientists, growers, consumers, we can all help each other to heal the planet. I want to thank you for your attention, for being here today and participating. And here's my email if you have any questions that you don't ask today and it came to you. And if you want to come and visit us at Rodale Institute in Pennsylvania, Kutztown, to see the vegetable systems trial and or to work with us, please let me know. And with that, I would like to look at the question and answer button here, and I'm gonna open the questions and see what we have. We have a question from Phyllis Rubin. What does mock mean in the graph? Yes, mock means that we took those inoculum and we sterilize it. And so every time we are comparing live inoculum of mycorrhizae as a treatment, you need to compare it to the same inoculum, but it is dead. And so we want to know if that makes any difference. So mock is a treatment that it is in statistics. We always need to include something that it is, if it is live versus dead, or if it is make a problem or no problem. So mock means that it is like a the same inoculums we use, but we kill them before we inoculate the seedlings. The second question came from Roger Williams. Please let us know in more detail the difference between black plastic versus reduced tillage. Definitely. This is the black plastic treatments usually that we prepare it according to the common standard that the growers usually use for growing vegetables. First, we prepare the bed by tilling down and incorporating the cover crop in the spring. We disc and pack and make the beds, and we cover the beds with plastic mulch. And during the season, we clean between the beds by cultivating if it is organic. And if it is conventional, we spray herbicide between the beds to control the weeds. So in contrary to that standard method that it requires a lot of cultivation and tilling and using a intensive equipment, we call it moldboard plow, we use another system, we call it regenerative or reduced tillage system. Because potato is a tuber and it is in the ground, it would be easier to use a different equipment when we call it reduced tillage by shallower tilling, chisel plow. But when we use for the squash, what we do, or when we used for the sweet corn, we rolled crimped with a roller crimper the cover crop, especially in the organic system. If it is in the conventional, it is still reduced tillage, but we burn it with a herbicide. So we added the toxic material that usually to kill the cover crop before we transplant. Hello, there is a question here. Hello, I would like to know more about the microbial summary discussed in the beginning. How does it matter if the microbial population different in different cropping system and management practices? Is one better than the other? It's a very good question. The microbes, each community, they work in a system across each other, and those could be helpful to see how they grow and they become abundant and at what level in the soil. Because those microbes, whether if they are bacteria or archaeal or fungal, they work to produce enzymes, they work to produce and recycle nutrients. So their factors, their abundance and communities, they change according to what we do with soil. If we apply toxic material, if we apply herbicides, if we apply any chemicals that reaches the soil, if we till the soil much more than needed, if we increase compaction, if we put synthetic fertilizers that are very high in ammonia, those organisms, their structures, their populations may change with time, but their production of the enzymes and the chemicals, they all recycle the nutrients to improve the plant growth. How they differ between cropping systems at this point, all what we trying to know if they are different. But as I discussed yesterday with Dr. Khan, we said we can try to look at the groups in each of those systems and see if they are similar and their ratios. And also we are gonna look how they could be, their abundance affect the nutrients that we have in the crops that we harvest. And also we are gonna look into the different soil health parameters that we have, such as aggregate stability, the bulk density, all those parameters, we are gonna put them all together and correlate them and see which of those factors really are close to those communities and affect the production, the yield of the nutrients. It is a big question with a lot of work to do in the future. But thank you for your question. Elliot says here, I have an off-topic question from the presentation. Does the RIFT training program go over some of the science involved with improving soil? Yes. The RIFT training program at Rodale Institute is a very comprehensive training program where usually they get information by learning in class, also going to the field to do the work, hands-on practices, visiting farms, and also they get introduced to all the science topics. The scientists at Rodale will give classes, like I would be giving classes on nutrient density, on the soil science. Others will be on weed management, on equipment, some others on compost, some others on livestock. So it is a very comprehensive program. It is we are very proud of this project, the RIFT program. It is expanding. It really helped younger generation to get back to soil, to nature, to understand how the science is really important to integrate by not only learning in class, but also when you are in the field. Phyllis Rubin has a question. Who decides what to study and design Rodale's VST research? Very good question, Phyllis. The design is based on collective information, collective decisions. Mainly at the beginning, we have a consortium where we contacted grower stakeholders, people who have all the experience in Pennsylvania and Mid-Atlantic, and selection of crops or the needs and what practices need to be compared to provide answers for and solutions. It is a combination from the scientists, from the grower stakeholders, the educators. And so this is scientists from Penn State. We have a nucleus that really help us to put those together. Now, as we go with the studies that we have year after year, is that the more depth that we want to go into, it requires more funding. And in that case, every time we are looking what is the fastest and easiest way to answer a question. Some of those will be short-term, some of them long-term, and they may require several years to get to that answer, but some of those will be equipped with even not only the VST, but satellite experiments to gain more information. Also, we try to work with growers to implement some of those practices to see how easy it is working or what challenges they have. And so there's a collective number of people who get into that. But the more we see things that we need to address and practices that need to be improved, we try to think how long it will take, what does it take. We put proposals, we work on those designs or proposals of questions we want to answer to help growers and consumers. I hope we answered all your questions here. If you still have anything, you are very welcome. Everyone who have questions, let me know if you have any more questions on your mind. You have my email here and you can ask. And also thank you for all the people who put on the chat recently. I'm checking that they gained information. It is a pleasure for me to see that this webinar was very informative. Thank you so much. And I leave you with the blessings for the day and every day. Thank you.